Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about bioinorganic chemistry. So in bioinorganic chemistry, first topic is elements in living system. You know the living matter is made up of 6 elements: carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur, which make up roughly 90% of dry weight of human body. Other physiologically significant elements like calcium, potassium, sodium, chlorine, magnesium, cobalt, iodine, zinc, fluorine, and molybdenum, and selenium. Their body composition varies greatly. For example, calcium make up about 2% of the body weight, whereas cobalt make up of only 0.0000004%. So, porphyrin ring. What is porphyrin ring? Friend, a porphyrin is a large rings molecule made up of 4 pyrols. You know the structure of pyrols, 5-member ring with 1 nitrogen. A smaller ring made up of 4 carbon and 1 nitrogen, which is heterocyclic compound. And these pyrrole molecules are linked by a chain of single and double bond, forming a huge ring. So imagine the pyrrole ring. A tetrapyrrole means as a compound is में 4 pyrrole को 4. Tetrapyrrole is the technical term for 4 pyrrole linked together. The ring is flat in space and has reasonably even distribution of electrons around its diameter. As a result, क्या होता है कि a porphyrin is classified as aromatic. A porphyrin molecule is highly stable in this shape. Porphine is the model of the universal porphyrin. So one pyrrole, another pyrrole attach with the methene group. Methene group means carbon, hydrogen with double bond. Now, if you see the vitamin B12 is produced when cobalt bind to porphyrin derivative. A deficiency in the vitamin can cause pernicious anemia and impair brain and nervous system function. And this porphyrin derivative pigment can be preferred to as the color of life. So this porphyrin ring gives the vitamin B12, you know the chlorophyll. If you add the magnesium, if you add in this ring nickel, then it become coenzyme, and if you add iron, then it become heme. So combination of porphyrin ring with different type of metal ion. So if you add iron 2 plus, then porphyrin ring become heme ring, metalloporphyrin ring, formation of heme group. So point is porphyrin is a macrocyclic. Structurally, porphyrin ring consists of 4 pyrrole ring for memory. Pyrrole ring is a 5-member heterocyclic compound consisting 1 nitrogen atom and 4 carbon atom. In porphyrin ring, 4 pyrrole ring connected with each other by methene groups. Porphyrin ring act as both basic as well as dibasic in nature. Porphyrin ring contains 11 conjugative double bond with a total of 26 pi electron, of which 18 pi electrons form a planar continuous cycle. Porphyrin ring structure is often described as aromatic. When porphyrin ring combined with any metal like iron, cobalt, or magnesium is known as metalloporphyrin. When Fe bind with the porphyrin, then it known as heme, which is compound in red blood cells, participation in the oxygen storage and transportation. The electronic transition in porphyrin ring is pi to pi star, and the complex with containing porphyrin ring color appears due to pi to p star electronic transitions. So in metalloporphyrin, what we are using? The role of iron, you know the living systems. What is the role of iron? So storage of iron like ferritin. What is ferritin? In French scientists discovered ferritin in 1937 when they extraction a novel protein from horse spleen that contains up to 23% iron by dry weight. Iron is stored mostly in ferritin, an iron storage protein. For the production of hemoglobin, myoglobin, and cytochrome, ferritin release iron is required. And this is important because unbound free iron is extremely toxic and causes cellular damage by promoting the productions of free radical in mammalian tissue such as liver, spleen, and bone marrow. Ferritin is a significant iron storage. Now, structure of ferritin. If you talk about the structure of ferritin, you will see the ferritin H and ferritin L and peroxidase activity and apoferritin shell. So transport of iron transferrin have the some key point. Transferrin bind iron is in plus 3 oxidation state. Fe2 plus is oxidation to Fe3 plus by ceruloplasmin, a blue copper protein. Transferrin bind copper 3 plus from stomach and enter into blood and transport Fe3 plus to bone marrow. In bone marrow, Fe3 plus is reduced to Fe2 plus and is delivered to ferritin before and after binding in transferrin. Fe is plus 2 oxidation state when bind to transferrin, Fe in plus 3 oxidation state. You know, transferrin deliver iron when it is reduced to Fe2 plus in bone marrow. Fe2 plus is further oxidation by ceruloplasmin peroxidase to Fe3 plus and pick up by the ferritin. Ferritin found in bone marrow, spleen, and liver. Ferritin contains two compounds. One is apoferritin, 24 protein chain each with the 175 amino acid. And second, iron core, it contains 4500 Fe3 plus iron. Ferritin release iron for the synthesis of hemoglobin, myoglobin, and cytochrome. So, transferrin is the best scavenger of iron in the animals. So, my friend, if you see the structure of siderophore, this is the one structure, a ferrichrome complex showing the iron binding with the three hydroxymate moiety. The structure of the cyclic peptide is shown by a circle. If you, I will show you this hydrophile later on. Now, I will talk the hemoglobin molecules. So, what is the biological role of hemoglobin? Hemoglobin is an oxygen carrier. Hemoglobin is a carbon dioxide carrier. Hemoglobin gives a red color to blood. Hemoglobin maintains the shape of the red blood cell. Hemoglobin acts as a buffer. Hemoglobin interacts with the other ligands. Hemoglobin degradation accumulates physiologically active catabolites. So heme plus globin, that is the hemoglobin. Now, if you talk about hemoglobin and myoglobin, so Bohr's effect, that is very important, and I will cover this question in my next video, and I will show you the structure also, so that this paper easily cover your concept.